A very big hello to my wonderful PU2 champions. I hope my dear children are doing super great. Well, I definitely understand that your FA2 examinations probably are gonna come. For some it might be ongoing, and in between that we have Brahmastra series. What's the point of all this thing? Well, I have taken a chapter that comes in your FA2 examination such that all your concepts, all your important questions are cleared, and that's my guarantee, that's my promise. If at all you watch this video, all the important concepts and questions of human health and diseases is going to be covered. Now this chapter is of super high weightage. If at all you have seen the weightage in boards, it gives you 11 marks. This is not a small chapter. Take it seriously and solve it. Over here, if at all you talk about the mark distribution, my dear children, it goes like this. You will get 3 1-marker questions, and that's the best part of the story. And it's easy. You can easily score marks over here. 3 marks easily. Now 1 3-marker and 1 5-marker will also be there. Together if you count, it will be 11 marks. Okay? And my dear children, I am putting forward a challenge in front of you. The challenge is to solve all the questions that I'm going to show over here with me. So how many questions do I have? Let me check, okay? Let me check. Don't get worried after seeing all this thing. It's for you guys. So 28 questions are there. So those of you who reach 28 questions out there have to comment down in the comment section that, ma'am, I have solved all the 28 questions, and I'm guaranteeing you, my dear children, with these 28 questions, you will be absolutely cool about this particular chapter. So, over here, the first set of questions would be taken from the model question papers of this year, 2025-26, such that everything in the paper is covered. So first we'll solve MQP1, then MQP2, then MQP3, then we'll go ahead with the question bank questions, the previous year questions and things like that. Now it might seem long for you, but when we start studying, it is going to be fun. Trust me on this, and if at all you believe me, do not forget to hit the like button right now. Starting with the first question that we have, human immunodeficiency virus replicates in. This is MQP1 question number 7. Human immunodeficiency virus means you're talking about HIV virus which cause AIDS. Now it has been asked, where does it replicate? Replicate means increase its number. Make copies. Where does it do that? Is it in B cells, B lymphocytes? Is it in NK cells? NK cells stand for natural killer cells. Okay, they are usually the ones which kills the cells which are affected with viruses. Okay, now helper T cells or PMNL. PMNL stands for polymorphonuclear leukocyte. Polymorphonuclear leukocyte. Leukocyte are what, my dear children? Leukocytes are nothing but the white blood cells. The other name of white blood cells is leukocyte. Now there is one such leukocyte which has different morphology of their nucleus, which is why you call it leukocytes. This cell's actual name is neutrophils. Now you are given all these options. You have to tell in which cell does the HIV virus actually replicate. The answer for this question is C, helper T cells. Why helper T cells? Because you know what, as the name suggests, helper T cells helps the other cells to become active, to basically increase the immunity and things like that. Now suppose I damage the mediator itself. Do you think the person who is helping will be there? No. Do you think immunity will still be strong? No. So that is why HIV virus targets, my dear children, helper T cells. You also need to understand one very, very important thing before we go ahead, and that is, initially when the HIV virus sneaks into our body, gets inside our body, it does not directly go to helper T cells. It basically searches for the easier resource, and that is macrophages. It goes to the macrophages. In the macrophages, they are going to increase their number, and now these HIV guys would be going and damaging our T helper cells, which is going to build up the immunity out there. Okay, so if at all you are asked, what is the HIV factory? And if at all you have given option of macrophage as well, please go with macrophages because it is known as HIV factory. This is where they initially come and develop. Later on, they go ahead, get inside the T helper cells and also replicate there. Okay, so if you have an option like macrophages, please go ahead with it and then do this. Now, if at all you are getting a question that HIV viruses majorly targets what? So it is the helper T cells. The major target is this only. But initially it just wanted to increase its number, that's why it went to macrophages. So if at all you're asked HIV factory, then macrophages. Okay. Or else if you just have helper T cells in the options, go with this, as in this case. Now that question is done. Question number 2. The drug which is depressant and slow down the body function is. It is a depressant, that means it slows down every functions of your body. It makes your mind go depressed a bit. Which is that drug? Is it smack? Is it crack? Is it hashish? Or is it charas? Always remember, crack is the other name for coke or what you call as cocaine. And cocaine is not a depressant, it is something that causes hallucination. It causes hallucination. You feel cow is flying, okay, and you are also flying. So that is hallucination which you get if at all there is someone who's taking cocaine, not you guys. You guys are not doing that, right? Don't do that. Now hashish and charas, my dear children, are different types of cannabinoids. That is a whole different type of drug. You call them cannabinoids. So cocaine is what? Cocaine comes from Erythroxylum coca. This is cannabinoid. Now smack, when you talk about it, it comes from a whole different plant which you call as Papaver somniferum. Have you heard about Tupperware? Okay, Tupperware bottles. Similarly, Papaver is over here. Papaver somniferum is there, which you also call as opium poppy. This is the plant from where you get smack. But ma'am, we didn't study about smack. We heard that, okay, from opium poppy we get 2 drugs. One is heroin and the other one which is there, it is morphine. But we have never heard about smack. So smack is the other name for heroin, my dear children. Smack is the other name for heroin, which is actually a depressant and slows down your body function. Okay, so smack you get from Papaver somniferum, or you call this as opium poppy, and crack is nothing but cocaine or coke which causes hallucination. Hashish or charas are cannabinoids, okay, which affects your cardiovascular system. Cannabinoids affects your cardiovascular system. Everyone has different effects. This one has this effect. So with one question you have understanding of 3 different things. Okay. Now over here I have also put, this is opium poppy or what you call as Papaver somniferum, and now the thing that you see is latex. The white color thing that you see is latex, and from this latex only you get the drug. So initially you get morphine. Morphine is the actual thing that you get, and you know what? Doctors use morphine not for themselves but for the patients who have underwent surgeries. Why? Because morphine which is there, it is a painkiller and sedative. It is a painkiller and it is sedative. That means when we have pain, ma'am, can we use this? No. It has to be monitored with the doctors only, otherwise, my dear children, it can also lead to death. So yes, that is there. So initially we had morphine from the latex. Now morphine undergoes a process which is known as acetylation, and that's how you get the diacetyl morphine, which is also known as heroin. So heroin has 3 names, chittu, bittu, kittu. So over here, diacetyl morphine, heroin, and also smack. My dear children, I am explaining all this thing in a way so that your concept is being understood. So heroin and this, everything you get from this, and also understand that heroin can be taken in the form of injection and also snorting, okay, and also snorting. So that are the 2 methods of taking this in. Now, and what effect does it have, if at all you are asked about that? It affects your central nervous system and also your gastrointestinal tract. Gastrointestinal tract. Opioids like them affects CNS and also gastrointestinal tract. Cannabinoids, cardiovascular system. Okay, chalo, next question. This is MQP1 question number 18. Symptoms of constipation, abdominal pain and cramps associated with the disease what? Over here you have got this option. So every third 1 marker is a fill in the blanks according to the model question papers. So amoebiasis, richness, nostoc, ovulation, colostrum and diversity. We all know there is only one disease given over here. So the answer has to be amoebiasis. Symptoms of constipation, abdominal pain and cramps are associated with the disease amoebiasis. It is caused by a protozoan whose name is ant amoeba. Ayyo, spelling is wrong, ma'am. Ant amoeba histolytica. Histolytica. It's a what? It's a protozoan. And my dear children, basically it affects your large intestine. And the major symptom which is not been mentioned over here is what? You get this thing, dysentery. So dysentery means when with stool blood also come, you call this as dysentery. So in this case, my dear children, people have dysentery also. It is why it is known as amoebic dysentery as well. Okay, done? Chalo. Now, question number 3. So every 3 marker question that I have seen in the model question paper says that this is a disease, tell what is the causative organism or probably tell the symptom of it. So you should know about all the diseases which are given in front of the chapter, go through it properly. Okay, most of it we will cover here itself. So name the causative organism of the following disease. Filariasis. Filariasis means what? Elephantiasis, where the person's leg becomes in the shape of an elephant's leg. Why, my dear children? Because there is accumulation of a fluid which you call as lymph fluid. There is no one to collect that fluid, because the one who collects that fluid in that particular vessel, this particular organism is there. It causes a block, which is why the fluid gets accumulated. Okay, it usually affects your lymphatic system, the lymphatic system which is there for collecting the extra lymph fluid out there. Always remember, and it is more towards the genital region and also the lower part of the body, which is why you have this. Now filariasis is caused by what? This guy what is this guy? Its name is Wuchereria bancrofti or Wuchereria malai. Okay, malai as in the thing that you get in milk, no, you can remember with the help of that. So Wuchereria, always remember capital W should be there. Wuchereria bancrofti is there, and apart from that W dot malai, small letter. The second letter should always be small, species name should be small. Okay, and they come under Aschelminthus. Aschelminthus means roundworms, everything they come under Aschelminthus. Now ringworm disease. Now you think that ringworm is caused by a worm. Basically ringworm disease is known like that because it makes a ring shaped structure. But it is actually a fungal disease. Okay, it is actually a fungal disease which is caused by microsporum. You just think microspore is there. Okay, so microsporum, or it can be trichophyton. This is all different types of fungus. Trichophyton. So there's a fighting happening between trichophyton and epidermophyton. I'm so sorry, there was a glitch. Okay, so let's be back again over here. So trichophyton, I remember, was fighting with something. It is fighting with another type of fungi which causes ringworm disease, and that is epidermophyton. So epidermophyton and trichophyton, they are fighting, and we have microsporum as well. All three of them can cause ringworm disease. Now talking about malaria, malaria is caused by another protozoan. Remember, we studied about a protozoan before also. In ant amoeba, histolytica is a protozoan, right? It causes amoebiasis or amoebic dysentery. Similarly, this particular protozoan causes malaria. The name of the protozoan is Plasmodium, and you have different types or basically species of Plasmodium. Over here I have a picture of all of them, I mean the written names. So malaria is caused by Plasmodium vivax, P. falciparum and P. malariae. Always remember, if at all you are asked which one causes the malignant or the deadliest form of malaria, it is none other than P. falciparum. Always remember, malaria itself is an important disease. You might get questions more related to this particular disorder, disease. Okay, fine. Can we go ahead? I hope this is clear. Please make sure you write the name of all the fungi, that is microsporum as well as epidermophyton as well. Chalo. Now the 5-marker question from MQP1, this is question number 38. Differentiate between innate immunity and acquired immunity. A, B, C part is there. So A part, differentiate between innate and acquired. So my dear children, when we are born with skin. We are born with tears and everything, right? What is this type of immunity? Does it provide immunity first of all? Skin? Is it providing any sort of immunity? Yes. If skin was not there, if at all you have seen, my dear children, things can easily get in and cause infection. So skin is a barrier. Okay, this is a type of immunity, or this is basically helping in immunity, which is why it comes under innate immunity. It is there with us from our birth. Now there is another type of immunity which is known as acquired immunity, something that we acquire throughout our lifetime. Suppose one day I got an infection and I got really weak. At this particular point of time, some new pathogen came into my body reacted to it by producing so many cells, but it didn't exactly know which one was actually working against the pathogen that just entered. Later on, when the same infection came back again inside my body was able to recognize it and basically stop the infection all of a sudden. So haven't I acquired this form of immunity in my lifetime when I had an infection? So this comes under acquired immunity. Okay, so innate is with us from our birth. Acquired immunity is something that we get through our lifetime. Fine. And also please remember, innate immunity, my dear children, would be non-specific. Your skin, does it say the virus you can get in, bacteria don't get in? Can it do like that? No, it is non-specific. It is same for all. It has the same action for all of these things that tries to get into our body. But acquired immunity, if at all you talk about it, it will be specific to particular antigens. For example, pathogen A came in my body will be releasing A group of cells to fight against this particular pathogen. B came in my body, then B group of cells will be probably fighting against this. So it is specific. A cannot go and fight with the B pathogen out there. It is very specific in nature. Now, innate immunity, does it have any memory? Does your skin has any memory that some infection has taken place? No. Does your body has any memory that has something like this taken place? In terms of immunity, my dear children, no. But whenever you talk about acquired immunity, there is a memory associated. Why? Because, my dear children, in acquired immunity, you basically have two types of cells which are playing the most important role, which is B lymphocytes and T lymphocytes. They are specific type of white blood cells that we develop in our body. Now, B lymphocytes, basically, whenever you see a pathogen coming inside, B lymphocyte will be producing antibodies against this particular pathogen. And not just that, they will produce some cells which are known as memory cells. What memory does it hold? It holds the memory that some infection has taken place earlier, and that time I was not able to produce enough amount of antibodies which were needed. Now this time I know what are the antibodies that I have to specifically produce in this case of infection. So that is a use of memory cells. Same thing with T lymphocytes. T lymphocytes, my dear children, they directly go and attack the cells which are there. Okay, they are direct ones. So here they produce antibodies, but here they directly go and attack. Apart from that, they also release memory cells which has the memory that this something like this has taken place, I have to do the same action again. So they have immunological memory if at all you talk about acquired immunity. Now example for innate immunity is skin, phagocytes, etc. So ma'am, you said that cells come under that category. No, certain cells are with us from our birth. Okay, it comes under cellular barriers, for example, macrophages, for example, natural killer cells, for example, PMNL, polymorphonuclear leukocytes, all of them, they are with us from our birth. Now, there are certain cells which are being formed later on, my dear children, and those are your B cells, your T cells. It happens slowly, and they do their action even more slowly, even only when you have an infection. So examples would be that. Now in your NCERT, which is one more thing which is important, is that what are the different barriers in the innate immunity? What are the different barriers which provides us innate immunity? There is physical barriers such as skin and everything. There is physiological barriers such as your secretions, for example, mucus that traps everything, right? Tears that also basically has enzymes such as lysozymes which will kill the bacteria. Now you have cellular barriers, as I told you, certain cells, and then there is cytokine barriers. There are certain cells which releases some other things which will be protecting the other cells out there. So I will just explain you that as well. So over here, suppose this cell is infected. It is infected from a virus. Now this virus infected cell will be releasing some very important material which is known as interferons, and tell the other cells that hey, listen, over here virus attack has been taken place, you have to take care of yourself. Okay, you have to take care of yourself. This is known as cytokine. Okay, this is cytokine and this is cytokine. This is example of cytokine barrier. All of them, they come under innate immunity. Cool. Now you have next question. Mention the cells involved in accomplishing the acquired immunity. So acquired immunity, I told you it will be always B and T cells. B lymphocytes and T lymphocytes are there for accomplishing the acquired immunity. Next thing that you have, refer to the below given diagram and answer the following questions. So here a diagram is being given of the what? Of the antibody. Now this diagram can be asked for you to make also. So you should know how to draw this particular diagram. Please understand, you should know to draw this particular diagram. And over here, if at all you are seeing properly, you have two long chains. What do you call these chains as? You call these chains as heavy chains. Okay, you call these chains as heavy chains. Now you see there are two small chains. You will be calling this chain as light chain. So heavy chain is 2 and light chain is also 2, which is why you say H2L2. Now over here, between the H2, two heavy chains, you see so many bonds. What are these bonds? These are actually sulfur bonds which are there. So there are sulfur bonds between two heavy chains. And there will be lesser number of bonds between a heavy chain and a light chain. And also understand, this is an antibody. What is the role of an antibody? To basically identify the antigens, the pathogens. So basically to a specific antibody, wherever a specific antibody is there, it will be reacting to a specific type of antigen also. It will be binding with specific type of antigens also. So where is this antigen going to bind? This is going to bind over here. You call this site as antigen binding site. Antigen binding site, and this is unique for all the different type of antibodies. How many antibodies do you have? Always remember GAMED, gamed, IgA, IgG, IgA, IgM, IgE and IgD. What is Ig, ma'am? Instagram? It's not Instagram, it is immunoglobulin. Immunoglobulin. So when I say that antibodies are produced, they are nothing but proteins, and the name of the protein is immunoglobulin G, immunoglobulin A, immunoglobulin M, immunoglobulin E, immunoglobulin D. Always remember that. And from your body, if at all the question comes that, ma'am, from the mother to the child, which is the first antibody to come, it is IgG. In the order, always remember, it is IgG which is transferred from the mother to the fetus through the placenta. Through the placenta, within the womb itself, it is being transferred. Now, ma'am, which is that particular antibody which is found in the mother's milk, the first milk which is known as colostrum. So basically in that you have IgA, A for Amma, you can remember like that. Okay, so first antibody to travel from mother to the child through placenta is G, and then when the baby is being born, A antibody is being given with the help of the milk. Now, name the molecules trapped in the lymph nodes responsible for the activation of lymphocytes that bind the A and B sites. So basically they are saying that there are some molecules which are trapped in the lymph nodes. They are trapped in the lymph nodes. That is a specific location in our body, okay, where the WBCs are very, very active. So over here certain molecules are trapped, and now they are saying that these molecules which are there, they are responsible for activation of lymphocytes that bind to the A and B site. That bind to the A and B site. So what will be this molecules, my dear children, which binds to the A and B site? It has to be an antigen. Antigen is going to go bind over there. So your answer for this question becomes what? Antigen. Now what does H2L2 in an antibody represent? I already told you, H stands for heavy chain, L stands for light chain. So two heavy chains and two light chains. So these are the information that you needed to know about what do you call the antibody thing. I hope it's clear. I hope you will remember. I'll also write placenta and milk over here so that you have a memory of that. And IgE which is there, it is produced during allergy. So allergy, IgE sounds the same, right? So you can remember like that. Okay, I might have taken a lot of your time, but I hope that with this you are gaining information about the whole chapter. Now name the cells involved in the humoral and cell mediated immune response. Now you told that in acquired immunity either the B cells are active or basically the T cells does the job. So whenever you're talking about B cells, what does B cell do? It produces antibodies. Where does this antibodies go? It goes and mixes with the blood, my dear children. And blood is what? Humor. Humor means fluid. Okay, so humoral immunity, that means in our fluid, there are so many cells. What are these cells coming from? Where are these cells coming from? What is their action? What do you call this as? So this is known as humoral immunity. Whenever the cells in the blood are responsible for our immunity, we say it is humoral immunity. And who is behind this? It is always B lymphocyte which is behind this. So this response is carried out by B lymphocytes. They produce antibodies that circulate in the blood and also the other body fluids. These antibodies bind to the pathogens and neutralizes them. Basically, when they bind to the pathogen, they might activate other cells to also come and eat them up. Okay, eat them as in pathogens up. Now, cell-mediated immune response, this response is carried out by T lymphocytes. Cell-mediated means it is going to be carried out by T lymphocytes. Because I told you, T lymphocytes, what is their job is that basically they can tell other cells to go and trap the organism, or basically they themselves can also go and kill the organism up there. So there are two types of, actually not two, there are multiple types of T lymphocytes, but majorly always remember there is helper T cells, and there is cytotoxic T cells. So cytotoxic T cells which are there, they directly go and kill the cells out there which is infected by a virus, cancer, anything out there. Now helper T cells gives the information to other cells, and now the other cells might be performing the same function. Okay, so cell-mediated immune response, this response is carried out by T lymphocytes. No antibodies are produced over here. Instead, T cells directly attack and destroy the infected cells or the cancer cells. Now, if at all you are asked, ma'am, there is graft rejection happening. Suppose there is organ transplantation happening, my dear children. This is known as graft. A graft is being put in your body, organ is being put in your body. Now it is being rejected. Who is responsible for this rejection? It is usually T lymphocytes. It says that this is a foreign thing, I cannot accept it, I will probably try to kill those cells out there. Okay, so which is why whenever we have a implantation or basically things like that happening, we are given immunosuppressants so that immune system is suppressed a bit. Cool? Cool. So this question was had come for visually challenged people as the C part of 5th question. So C part of the 5th question was changed for visually challenged people, and this was the question I thought I should discuss. I discussed. Now, going ahead with the next question, this is from MQP2 and question number 7. So, antivenom given against snake poison contains what? So, when we suppose someone have got bitten by a snake. Now, when they go to the hospital, doctor does he say that, he or she say that, hey, let's wait for our body to produce antibodies against it? No, right? So, usually what happens is that basically the thing that is given, within that only you will get antibodies, ready-made antibodies, because you cannot rely on your body to produce antibodies at that particular point of time. Now just wait for a quick second, okay? Yes, so we cannot rely on our body, which is exactly why antibodies are given. Because the production of antibodies might take a huge lot of time, my dear children. And over here, so snake venom antiserum is given. This is the medicine that is being put down into the patient. So it contains just the antibodies against the venom. So you might be thinking, where do we get the antibodies? So the venom for the snake is being collected. Similarly, antibodies which are being produced by the people who have been bitten, that is also being collected, and that is being synthesized artificially as well. And that's how we get the antibodies. Okay, and this type of immunization, what do you call this type of immunization? Where you are giving ready-made antibodies. We are not waiting for your body to produce, you are giving ready-made antibodies. What do you call this type of immunization as? This is known as passive immunization, or this is a type of passive immunity where you are being given ready-made antibodies. If at all your body is producing antibodies by itself, it is known as active immunity. But here we are relying on passive form of immunization. Cool? Cool. Now, question number 7. The substance produced by a cell in viral infection that can protect other cells from further infection is, this is again model question paper 2 question number 8. So what is that substance produced by a cell in viral infection that can protect the other cells? So as I said, one cell is affected, it is producing something that protects the other cells. What is this thing that is being produced? That is the question. I already explained you while I was talking about innate immunity. It is interferon. It is interferon which is being produced. So over here you can see the picture. The virus is there affecting this particular cell. Now this cell releases interferons. Now this interferon, basically it gives signals to the neighboring uninfected cells to destroy the RNA and reduce the protein synthesis, or probably it can instruct the cells to undergo apoptosis, suicide also, so that they are not affected. Because suppose if they are affected, my dear children, they are giving space for the viruses to replicate. That is the reason. Or probably they can give the signal to activate the other immune cells and tell that there is a problem that has taken place up over here. So always remember, this comes under cytokine barrier of innate immunity. Okay, it's there with us from our birth. So that is also sorted. Now again you have a fill in the blanks question. Dash is a very effective sedative and a painkiller. So we studied about the depressant and who slows down our body, that was heroin. And at that particular point of time I told you doctors give sedative and painkiller to patients who have underwent surgery. What is that particular drug? It is none other than morphine. From morphine only you get heroin, right? So morphine is the answer for this particular question. Okay, it's an easy question, but sometimes we forget it since we have a lot of things to study, right? Now question number 3, here I've given you the answer. Please not see the answer now. So question number 10, not 3. So question number 32 of your model question paper 2. Mention the causative organism for the following disease. Ascariasis. Ascariasis is caused by Ascaris lumbricoides. As you can see over here, it is a roundworm. So in the same family comes your next Wuchereria bancrofti, remember, which was causing filariasis. It's again given. In next model question paper also it is given. So filariasis is caused by Wuchereria bancrofti or Wuchereria malai, and Ascariasis is caused by Ascaris lumbricoides. Now typhoid, it is caused by what? It is caused by a bacteria. They are not coming under Aschelminthus and things like that. This is a whole different category. It's a bacterial disease. It's a bacterial disease. So typhoid is caused by Salmonella typhi, a bacterium. Salmonella typhi, which is a bacterium. Okay, typhoid is caused by Salmonella typhi, please remember that. And what symptoms do you have in typhoid? You have high sustained fever that occurs, okay, that occurs whenever typhoid is getting serious. So please remember those small things as well. Now, again we have landed to the 5-marker session. Model question paper 2, question number 43. The flow chart shows replication of retrovirus in the host. Answer the questions given below. So flow chart shows what? Replication of retrovirus in the host. What is a retrovirus? HIV is a retrovirus. What does it cause? It causes HIV/AIDS. So before filling this particular thing up, I need you to go to the last question, because the last question has something to do with this. So over here I have a question. Give the schematic representation of replication of a retrovirus. This is PYQ question. It came in March 2022. It has also been repeated, but yes, I'll tell you. So over here, always remember, virus which is there, it will have RNA as a genetic material. The HIV virus which is there, it has RNA as a genetic material. It has 2 strands of RNA. Both of them, they are not combined together. They are not double-stranded. Both of them are single-stranded. They are there. 2 of them are there. Okay. Now what happens is that virus, it comes and infects the normal cell which is there. Usually it is a macrophage which is initially being infected. Anyways, so it is coming and infecting a cell. So virus infects a normal cell. Now once virus infects a normal cell, what is going to do? It is going to transfer its RNA inside the cell. Ma'am, why is it this particular thing happening? So my dear children, we have studied in molecular base of inheritance that from DNA we get RNA. From RNA we get protein, right? So here they have the RNA, and our cells, it's our cells, right? Our cells will be having the DNA. RNA is introduced, we have DNA. If at all this particular guy wants to use our cells machinery, it has to make sure that it is not RNA which is there, but rather it should form its own DNA. Can it do that? Yes. So viral RNA, after being introduced into the cell, you know what? Viruses, they carry a very important enzyme with them which is known as reverse transcriptase. So usually we say transcription is DNA to RNA. If at all we are reversing the process, this is known as reverse transcription. And for this enzyme, you have an enzyme reverse transcriptase which reverses the whole process. Now you have the viral DNA being formed. This viral DNA incorporates into the host genome. It goes and gets inside our genome, that is host genome. And now when it gets incorporated over there, it uses our machinery to basically what? Replicate, to do all the other functions out there. And basically now they will produce new viral RNA in the infected cell. So with the machinery, they made their own RNA. Okay, they made their RNA, and this RNA and everything, my dear, apart from that, they made their proteins also. Proteins like these enzymes which are there, they are proteins. You need enzyme to do reverse transcription. You need enzyme to get into our DNA, to get to be a part of our DNA as well. You need enzymes to basically later on cut down the small, big proteins which have been formed into small components. Everything has been made with the help of our machinery. So new viral RNA is produced along with all the other proteins, and now all of them assemble to form a whole new virus, and now these viruses will move out of the cell and go infect the other cells. This is how they work. This is how HIV virus works. So initial factory of HIV is known as macrophages, and now when they have left macrophages, they go to the T helper cells and basically cause problem out there in the T helper cells. Okay, and that causes our immunity to go low. So you might be asked to make this diagram properly. Okay, if at all you are asked that, do not mug it up, rather remember what exact things are happening over here. And as I said, this question has come. Now a related, very related question is over here which we were discussing. Where is that? Yes, it's here. Fill in the missing data in boxes labeled as 1 and 2. So let's see what is 1 and 2 over here. So retrovirus, virus infects the normal cell. Viral RNA is introduced into the host cell, and now viral RNA undergoes reverse transcription. After reverse transcription, what is going to happen, my dear children? Viral DNA is going to be formed. Viral DNA is going to be formed. Now viral genetic material in the nucleus of the host cell. Now the viral DNA got into the nucleus of the host cell. Then viral DNA incorporates into the host genome. It gets inside our genome as well. Now what happens? Because after that, virus comes out of the host cell. So after this, it will produce its own RNA, it will produce its own proteins, everything. Here it is said that viral DNA incorporates into the host genome, and then what does it do? It basically produces viral RNA and protein, and now later when they are all assembled, virus comes out of the host cell, and basically new viruses would be affecting the other cells. So fill in the missing data. Box 1, viral DNA is formed, and box 2, viral components such as proteins and RNA synthesized, and the assembly of new viruses also happen. Either of them, but this is the first thing that you need to write. Okay, this is the first thing that you need to write. I hope now it's easier for you when I explained you the other one. Now give an example for retrovirus. Ma'am, we all know it is HIV, you have told it. But when you write HIV, make sure you're writing the entire name, human immunodeficiency virus. Okay. Now next thing, name the disease caused by this retrovirus. What is the disease which is caused by this particular retrovirus? It causes AIDS. Again, you should be knowing the full form it. Acquired immunodeficiency syndrome. We are becoming immune deficient. Okay, we acquired some disease which makes us immune deficient, and you have lot of symptoms of this. That is what you mean by syndrome. So acquired immune deficiency syndrome, or you call this as AIDS. Now name the diagnostic test for this particular disease. What test do you have to diagnose HIV? It is ELISA. It is ELISA. ELISA stands for enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay. How does it work? I can give you an example. Suppose we already know that in the patients who have HIV, they produce this antibody. They produce this particular antibody. So basically we have some setup where the antibodies are already present. Now suppose I have a doubt with a person that the A person has HIV. Let's take the sample of them. In their sample, obviously you will get antigens. Suppose you get antigens. Okay, antigens, that basically you're talking about virus, basically the one that causes the particular disease. If at all the antigens are there, they will specifically go and bind over here. If they are not specific to this, if the antigens are not coming and binding, that means basically they are not related. But over here, we got the antibodies from a patient who had HIV/AIDS, right? So we know this antibody is specific to antigens of the HIV/AIDS virus. So over here, if at all it matches, we definitely know that that particular person has disease. Now how do you see it? You take help of another antibody. this antibody which comes through, my dear children, and comes and attaches over here, this is linked to an enzyme. It is linked to an enzyme, which is why you call enzyme linked. And now what do? You put some coloring substance. When this chromogen coloring substance is being put, and when in the presence of this enzyme, they will change the color and give a different color. Give a color, if at all this whole thing has taken place. So that is why you call this as enzyme linked immunosorbent assay. Okay, so initially you can take a antibody over here, or probably you can take an antigen also, but for that we need to go in depth. We don't need to do that as of now. Please remember, it is just ELISA. Now one very important thing, you will be asked this question as well. You may be asked this question as well: how is HIV transferred? So you should be able to answer to that question through through patients who are undergoing repeated blood transfusion. They can have it if someone uses injections for drugs and things like that, and they do not take care properly, then obviously it can pass through like that also. And organ transplantation, blood transfusion, as I already told you. Apart from that, physical sexual contact with a person who is having AIDS, that can also lead to this. From mother to child also transmission can be happening. Okay, so all of this, please remember that. Now next question. Question number 12, MQP3, question number 7. Okay, so now we have reached model question paper 3. Which of the following plants does not possess hallucinogenic properties? Hallucinogenic means, that means it does not give an idea of flower, the cows are flying and things like that. Okay, hallucinogenic means the one that causes hallucination, and here it is being asked the plant that does not possess hallucinogenic properties. So what is the one that we studied earlier that causes hallucination? It was cocaine, and cocaine you get from Erythroxylum coca. Remember, you get it from Erythroxylum coca. What about Atropa belladonna? It also causes hallucination. What about Datura? It also causes hallucination. 3 of them are there given in your NCERT that causes hallucination. These are the 3. Erythroxylum coca produces cocaine. Please remember that. Now over here, you have Papaver somniferum. Papaver somniferum, heroin and morphine are the drugs that you get from it. And have you studied that it causes hallucination? No, my dear children, it does not cause hallucination. Always remember, you have 3 types of drugs: opioids, okay, and cannabinoids. And over here you have the Erythroxylum coca. So basically, coca alkaloids. Coca alkaloids. This is going to affect your central nervous system, that's your brain and your spinal cord, and also your gastrointestinal tract. Okay, cannabinoids which are there, they are going to affect your cardiovascular system, that means your heart and everything. Cardiovascular system. Coca alkaloids, they will affect your CNS, central nervous system specifically, and in CNS they are going to affect the secretion of dopamine. You know, dopamine is a neurotransmitter secreted from one neuron to the next, between the next neuron. It makes us happy. Dopamine is a happy hormone. Now, what does it do is that it basically makes the dopamine to be in continuous motion. Dopamine is being released, which is why we get a sense of euphoria. We are intensely happy, and also it leads to hallucination. So this is the thing. And always remember, if at all you get a question that there is some drug which basically increases the heart rate. Okay, heart rate, blood pressure, everything. So you might think, ma'am, cardiovascular thing was cannabinoids. No, so the answer for that will be cannabinoids. But the answer for blood pressure, increasing of blood pressure and heart rate, is not cannabinoids. Rather, it is nicotine. The thing that you get from smoking. Nicotine causes increase of blood pressure. Okay, so please have this thing in your head. It is super useful. Opioids example are heroin and morphine. Cannabinoids is charas, ganja, marijuana, everything out there, okay, which you can probably remember, bhang, everything. Coca alkaloids example is cocaine, and the other 2 things that is there, that is Atropa belladonna and also Datura. Okay, apart from it, you have 2 more drugs given in NCERT: barbiturates, amphetamines. Okay, they are the ones which are basically given to patients who have depression and things like that. So that is barbiturates, amphetamines and things like that. Okay, you might come across some questions, or you may not, I do not remember exactly, but yes, Please have that understanding of all the drugs. Now choose the correct statements. This is question number 8. Innate immunity is accomplished by providing different types of barrier. I told you, yes, there are different types of barriers. So this is correct statement. Now acquired immunity is present from birth and is inherited from parents. Do we have it from birth? Wrong. Acquired immunity can be divided into antibody mediated and cell mediated. Yes, antibody mediated means humoral immunity, that means B cells, and cell mediated means T cells are the ones. So this is correct. Innate immunity is also called specific immunity. It is not at all specific. Acquired immunity consists of specialized cells, that is B cells and T cells, and antibodies that circulates in the blood. Yes, it does. So basically, can I say 1, 3 and 5 are correct? 1, 3 and 5 are correct. That means your answer is C option. Your answer would be C option. Okay. So yeah, since I was talking about it, I feel now I should be writing them also. So over here, barbiturates as well as amphetamines. Amphetamines, they basically treat insomnia. That means if at all you are not able to sleep and all, that is known as insomnia, and also depression. Insomnia and depression are being treated with the help of barbiturates and amphetamines. Okay, now if at all you are given a question that which is the drug that is being abused by the sports people, it is cannabinoids. Cannabinoids is the drug abused by sports people, as in they use it. Okay, they use it very much. Sports people. Cool. I think I'm able to give you answers for a lot of questions from in this particular slide itself. You might be seeing some questions. You will get to know what I was trying to tell. Okay. Now question number 18th. Dash are the sites where immature lymphocytes differentiate into antigen sensitive lymphocytes. This we have not discussed until now. Now immature lymphocytes means basically, my dear children, the B cells which are being produced. The B cells which are being produced, they do not know what to do, what are the antigens and things like that. They need to be taught, they need to be matured. So there is a place where they get matured. Okay, B cells, they mature at one particular place. T cells, they mature at another particular place. So what is the site known as altogether? I told you there are 2 different sites, but do you have any general term used for it? Yes, you call them primary lymphoid organs. So primary lymphoid organs are the place where the lymphocytes basically they are produced, they get matured to what? To become antigen sensitive. They will know that this is the antigen that they have to react to. But they don't start doing the action here. Always remember, they do not do distribution over here. For that, they have to travel to next type of organs which are known as secondary lymphoid organs. Okay, which are known as secondary lymphoid organs. So over here, they just become antigen sensitive. They just become antigen sensitive. This is also very important question. And what is the example for primary lymphoid organs? You have 2 of them which you need to remember. One is bone marrow and the other one is thymus. Bone marrow and thymus. Bone marrow is there within each of our bones, yeah, and thymus is basically located over here beneath the sternum, the rib bone out there. Okay, so bone marrow and thymus is there. Bone marrow is the place where all of our body's blood cells are being formed, my dear children, and one such cell is B lymphocyte, another cell is T lymphocytes. Both of them are born there. B lymphocytes mature in the bone marrow itself, but T lymphocytes goes to the thymus for maturing, which is why you say that both of them are primary lymphoid organs where they become antigen sensitive. Remember, they are not going to be the effector cells. Effector cells means they are going to do dishum. That does not happen here. That happens in secondary lymphoid organs, such as spleen, such as lymph nodes, such as Peyer's patch, about which we will study later. Chalo. Again a question, as I told you, all the 3 marker questions have come from the causative organism, and this is the exact question, they just moved here and there. So question number 32, Ascariasis is caused by Ascaris lumbricoides, Aschelminthes. Okay, please know the symptoms of all these diseases, super important. Ringworm is basically a fungal disease caused by all this type of fungus. Now typhoid is caused by bacteria Salmonella typhi. Okay, children, do you want me to make a list of all these like bacterial diseases, this disease, causative organism, symptoms and everything all put together? If at all you want me to do that, let me know in the comment section. Okay. Now, question number 16. MQP question number 39. So this is not a 3 marker, this is a 5 marker. Name the group of genes that have been identified in normal cells that could lead to cancer. So they are asking you that there are a group of genes which are found in our normal cells, but these genes can lead to cancer, can make a cell cancerous. Do you know the name of these genes? They are known as proto-oncogenes. Proto-oncogenes. So usually they do their own function of producing some proteins and things like that. Now suppose there are some activations, some mutations happening over here, they turn out to be cancerous in nature. They can make the cell a cancer cell. So what is the name of the genes? It is proto-oncogenes. Similarly, you have some viruses. You might have heard about viral oncogenes. There are some viruses which transfer their genetic material into ours, and now when it is transferred, it causes cancer. So that is known as viral oncogenes. So this is there within our body, all of our cells, we have it, proto-oncogenes. Now when they mutate or they become active, we will get cancer. Now, which techniques are useful in detecting cancers of internal organs? Which is a technique that is used for detection of cancer of internal organs, my dear children? For internal organs, you need CT scan. What do you need? You need computed tomography. But here you should not be just talking about one particular detection, rather you talk about all of the things. You need to talk about all of the different methods of detecting cancer. Why? Because this is a 3 marker question, as you can clearly see. Okay, as you can clearly see. Then you can probably rely on to other things. So over here, the 3 things is biopsy and histopathology. First thing, suppose there is a lump over here. I take out a bit of the tissues, that is known as biopsy, and when I do the testing, this is known as histopathology. So microscopic examination of tissue to detect malignant cells, that is there. Apart from that, CT scan. Now this is the most important thing, which basically gives an idea of the 3D structure of an internal organ. So computer tomography uses X-rays. Uses what? X-rays, ionizing radiation, the one that is not healthy for our body. Uses ionizing radiation or X-rays to generate 3D images of internal organs to detect what? Tumors. Now MRI is there, magnetic resonance imaging. Over here you are going to use magnetic fields and non-ionizing radiations. Non-ionizing radiation is fine for us, okay, comparatively fine. Now magnetic fields and non-ionizing radiation to accurately detect pathological and physiological changes in the living tissue. In the living tissue, if there are some changes, that is being detected by MRI. So 3 of them, 3 different things. Remember CT scan with 3D structure of internal organs. Remember MRI with scanning of our living tissue. Always remember that, okay? Now there is one more question. Why are cancer patients often given alpha interferons as a part of the treatment? The interferons which were released before, that is different. This is alpha interferons which you're talking about. Why do you think cancer patients are given alpha interferons? Because they are biological response modifiers. They are biological response modifiers. They increase our immunity. That is the use of alpha interferons. Alpha interferons are biological response modifiers. They activate the immune system and helps boost the body's ability to destroy the cancer cell. It enhances the immune response. Okay, so these are things. Cancer is one of the very important topic of this particular chapter. Whenever we talk about diseases, usually it's cancer which is being asked a lot of times. They talk about what are the features of a cancerous cells and all of this thing. So make sure you pay attention to it. Now my dear children, if at all you have reached until this point, and if you think this lecture had been helpful for you, I tell you that basically there are 3 videos being put up, and I majorly put these 2 videos for my subject because I know that these chapters were coming for your FA2. So if at all you have not watched them, please go and watch them. It's for you guys. If you want to watch it, you can. Okay? Now, we completed the model question paper questions. We are going to go ahead with the revised question bank questions. Okay, revised question bank questions and the PYQ questions and the previous year MQP questions. So you will find more of MCQs, okay, and we will study with the help of them. Now question bank question number 5. Which of the following pairs correctly matches a disease and a pathogen causing it? So you have to tell the correct pair. So typhoid is caused by Salmonella typhi. It's a bacteria. Is it correct? Yes, ma'am. Pneumonia is also caused by Haemophilus pneumonia. Haemophilus was there. So yes, this is correct. This is also correct. Is it correct? No. It was Haemophilus influenzae which was causing pneumonia, and Streptococcus pneumoniae which was causing pneumonia. Two different bacteria were there which were causing pneumonia, but here they have mixed the both of them. So this is wrong. Malaria is caused by Plasmodium. Ascaris lumbricoides causes what? Ascariasis. Ringworm is caused by a fungus. Here Entamoeba histolytica is being mentioned. It causes amoebiasis, which is why your only answer for this question becomes A. Now how do you remember the concepts of this particular chapter? By solving questions. As of now, I think you are realizing that when you solve so many questions with the same things, you are able to remember things better, right? So please apply the same logic and try to give your best. Now hemozoin is a, do you know what is hemozoin? Hemozoin is what? So my dear children, I'll tell you the story of Plasmodium. The Plasmodium which causes malaria. So the answer for this question is toxin released from Plasmodium infected cell, but you need to know how and what is it. So over here, suppose Plasmodium is there, and Plasmodium, as I told you, it's a protozoan that causes malaria, right? It is spread through a vector. It is spread through a vector whose name is Anopheles mosquito, the female Anopheles mosquito. So female Anopheles mosquito will be having a stage of this Plasmodium which is known as sporozoite stage. Sporozoite stage is there in the salivary gland of the Anopheles mosquito. Suppose this Anopheles mosquito came into my body and tried to suck my blood. When it does, it has to make sure that my blood is not getting clotted, because in my blood there are so many things that will make a clot. In that case, the mosquito will not be able to drink the blood, which is why it releases its saliva, because in its saliva there are anti-clotting enzymes. But when it passes its saliva, isn't it also passing the sporozoites? Yes, ma'am. Now sporozoites are successfully inside the human body. Now in human body, it has two camps. They're terrorists, they have two camps. First camp is our liver. The cells of our liver are known as hepatocytes. Okay, in our liver they come and they basically undergo asexual method of reproduction, and later on when they are too much in number, what will happen? Basically the hepatocyte or these cells of the liver, they are going to get broken open. Okay, they are going to get lysed, which is why all the small organisms, the Plasmodium which was there, they will get into our blood now. They are properly going to into our blood now. In our blood, they take the second camp, which is our RBC, erythrocytes or red blood cells. In our RBC also they do the same thing. They undergo asexual reproduction, and the RBC get lysed, and basically what will happen? Basically again they will be released in the blood. Now you have to make sure of two things which is happening over here. Within the RBC, when they are undergoing asexual reproduction, they are forming into different type of Plasmodium. They have different stages, okay? One such stage is gametocyte. So they form the gametocyte state. What do you mean by gametocyte? Gametocytes are the one that can form the gametes. And the gametes, when they fuse, you call it sexual reproduction. The gametes, when they fuse, what happens, my dear children? It gives rise to a new organism out there, right? So similarly over here, gametocytes are being formed in our body. And now when the cells blast, the gametocytes are released in our blood also. But do you think gamete formation happens in our body? Do you think fusion happens in our body? No, it does not happen. So now it is in our blood. Okay, now it is in our blood. Another mosquito, Anopheles mosquito, comes and drinks it. Basically, it takes the gametocytes within its body. Now in the gut of the mosquito, within the gut of the mosquito, these gametocytes will form gametes. The gametes will fuse, and again the cycle will repeat. There will be a sporozoite stage which comes into the salivary gland, and the same thing happens. Ma'am, in between all of this, you didn't tell about hemozoin. Listen to me carefully. In RBC, when they were undergoing asexual reproduction and producing gametocytes, my dear children, there is one more thing that is happening. You know, Plasmodium feeds on our hemoglobin molecule, on hemoglobin. So over here, the hemoglobin is being broken down to two things. One is heme protein and the other one is globin. Heme protein and globin. Now what happens to heme is that heme is kind of toxic for this particular plasmodium. So within the cell they were doing all this thing, and here you have the plasmodium also. It is very toxic for them to stand by heme. So they convert the heme into something else which is known as hemozoin. What do you call it? Hemozoin. So hemozoin is now being produced by them. They're converting heme to hemozoin to make it less toxic out there. Now the problem with this is that when the cell is going to lyse because of this plasmodium, when they undergo asexual reproduction and produce gametocytes, they also release hemozoin into our blood, and hemozoin is toxic for us. That is why we get what? Fevers and chills. So in malarial patients you will find fever and chills happening because of the same reason. Okay. Now from here, question which can come is in which stage does it affect a human being? It is sporozoite stage. Does sexual reproduction happen in the case of our body? The answer is no. Where is gametocytes produced in the case of plasmodium? Gametocytes are produced in our RBC. Okay, always remember these three questions. Super important. So hemozoin is what? Toxin released from plasmodium infected cells. C is your answer for this particular question. Done. Now, question number 19. Let's solve this. Which of the following is an autoimmune disease? Autoimmune disease means what? Basically, our body does not know it's our own cells that is being destroyed. Our body is destroying its own cells. This is known as autoimmune disorders or diseases. You have rheumatoid arthritis, you have tuberculosis, you have malaria, you have AIDS. AIDS, you know, is caused by a virus. Malaria, you know, is caused by plasmodium. Tuberculosis, TB is caused by bacteria again. But my dear children, over here you're talking about autoimmune disease where your own cells are going to destroy your own cells. The answer for this question is rheumatoid arthritis. Okay, it is not able to differentiate between self and non-self. Over here, the cells which are there in our joints are being destroyed, which is why we have joint pain and things like that. So your answer for this question is A, rheumatoid arthritis. Okay. Next, the site where lymphocytes interact with antigens and proliferate to become effector cells. I told you. Lymphocytes basically are now going to see the antigens, they are going to do dishum. They are going to become effector cells. What are these sites known as? These sites are known as secondary lymphoid organs. Secondary lymphoid organs. So over here, thymus and lymph nodes is given. Thymus, you know, is a primary lymphoid organ, so wrong. Bone marrow and thymus, both of them are primary lymphoid organs, so wrong. Peyer's patch and tonsils. This is the answer. Bone marrow again is wrong over here. So Peyer's patch, tonsils, spleen, all of them are secondary lymphoid organs. So you might have studied, it might have happened with you guys that whenever you get an infection, basically you get tonsillitis. Why? Because over here you have so many WBCs which fight with the antigens, which is why there is an inflammation being caused. Okay, so our tonsils is a secondary lymphoid organs. Peyer's patch is found in the lining of our intestine. Peyer's patch is found in the lining of the intestine, and spleen, we will talk about it later, it's near to our stomach. It's also a secondary lymphoid organ. Okay. Now, 21st, injection of antitoxin in tetanus confers which type of immunization? I am giving antitoxic for tetanus. Why? What type of immunization is this? I am giving ready-made antibodies. Ready-made antibodies means, is my body actively participating in immunity? No, it is passive immunization, my dear children. This is known as passive immunization. You can be asked a question regarding what is the difference between active and passive immunity. What is the difference between active and passive? Active means you are relying on your own body to release antibodies. Passive means you are giving ready-made antibodies. In the case of active, basically it is slow, but it will be providing more immunity to me for a longer time period, whereas in the case of passive immunity, I'm just giving antibodies, which is why until when the antibodies are there, I will be immunized. So it is temporary in the case of passive immunization. Okay, in active immunity, do you have memory? Yes, because my B and T cells both will be involved. In passive immunity, do we have memory cells? I have been given antibodies, so how will I be having memory cells? That is the difference. Example example, active immunity, I get an infection and basically my body develops resistance against it. So that is active immunity. Passive immunity means from the mother to the child you are giving milk which has IgA. This is example of passive immunity. This is example of passive immunization. Okay, when the process is being done. Vaccination, in which category does it come in, active or passive? In vaccination, what do? You give dead or live attenuated form. That means something that does not causes any problem in us. You are just giving it into the body such that your body can react to it, so that your body can produce its own antibodies. So that is active immunization. Vaccination comes under active immunization, which is why in COVID we were given vaccination, even though they have so many troubles around, but yes. So we are given vaccination from the point we are born, polio virus, everything, for that we get vaccination, right? So what is this vaccination? We have been told that this is something that we are inserting in the body and our body will react to it. Next time if something like this will come and attack, make sure you produce even more antibodies. That is the logic. So active immunization is happening in the case of vaccination. Okay, whereas over here it is passive immunization because you are giving ready-made antibodies. Cool? Cool. Now, tobacco consumption is known to stimulate secretion of adrenaline and noradrenaline. The component causing this could be, so tobacco which is used in smoking, my dear children, it is stimulating the secretion of two very important hormones, adrenaline and noradrenaline. You know what? They are responsible for fight or flight response. Either you fight or you run response. So they obviously increase your blood pressure and everything. So the component that is used in tobacco which causes all this thing is what? I already told you, BP, heart rate increase. It has something to do with nicotine. Nicotine is the answer for this particular question. And with that, I might forget it, which is why I'm telling you here itself. There are things, so what happens when a female takes drug? Sports person, I'm talking about sports person. And also what happens when a male take drug? Please write the answer in the comment section. This is your homework and this is a very important question. Okay, when a female sports person takes a drug, what happens? What happens when a male sports person takes a drug? It is given towards the end of the chapter, super important. Okay, now again the question. Okay, the question number has been repeated. Okay, charas and ganja are the drugs which affect. Charas and ganja, marijuana, bhang, everything I told you comes under what? Cannabinoids. And cannabinoids, my dear children, affects our cardiovascular system. Affects our cardiovascular system, and the sports people basically they use it the most. Okay, that is what is said. Now, it's not said by me, so please forgive me. It's said in the NCERT. Okay, cannabinoids. They are cannabinoids. Cool. A chemical structure is given below. Identify its plant source. What is this structure? Now, always I'll tell you, this looks like an O. This looks like an O, that means it's an opioid. Well, which is the opioid that we know? It is morphine. It is actually the structure of morphine. So, we have two opioids, morphine and heroin, but first morphine. So, you get this from papaver somniferum. If at all, my dear children, you see something like this in the structure, it is none other than a cannabinoid. So there are two pictures given. One is this O-shaped structure, which is opioids, and one is cannabinoids. Okay, remember it. The disease chikungunya is transmitted by, ma'am, we studied about all the diseases, but chikungunya, when did it came? So chikungunya, my dear children, it is a viral disease. Basically, it is being transferred with the help of mosquito, which is Aedes mosquito. So it's not just chikungunya but also dengue. Dengue is also caused because of the bite of Aedes mosquito. So you have three mosquitoes to remember. One is Anopheles, this is Aedes, and the third one is Culex. All of them females. Okay, male mosquitoes does not take our blood. So this causes malaria. This causes dengue and chikungunya, and this causes basically, remember, filariasis, elephantiasis. It is caused by what do you call filarial worm, that is Vicariae bancrofti. But who transfers it? Who is the vector? The vector is what? The vector is what? The vector is Culex mosquito. So this is for filariasis. And over here, please remember one more very important thing I forgot. Yeah, Anopheles mosquito, it is a carrier and also a host. It carries, but also it is a host because in its body also things are happening, right? It is undergoing division. So it is also a host and also a carrier. So in NCERT, these small things are there. Okay, tell the name of a viral infected disease. So polio is a viral infected disease. Okay, so please remember all this small information. It's not just common cold which is caused by a virus. And there is a question, what is the difference between pneumonia and common cold? In pneumonia, my dear children, in pneumonia, your lower respiratory tract is being affected. In common cold, your upper respiratory tract is being affected. Your lungs and all is not being affected because of common cold. Always remember that. Cool? Chalo. Now, 25th question. Identify the enzyme which deviates from the central dogma of genetics during HIV infection. Central dogma of genetics is what? DNA, RNA, protein. This is central dogma of genetics, and DNA forms other DNA with the help of replication. Now, which is that enzyme that deviates, that moves away from the central dogma of genetics during HIV infection? It will be reverse transcriptase, because over here from RNA you get DNA. So this is the answer. Okay, this is the answer. I told you there are multiple proteins. One protein is reverse transcriptase, another is integrase, which lets the viral DNA incorporate into our DNA. Integrase helps in integration. Remember like that. Chalo. Now MCQ questions which have been mostly repeated have been done. Now we'll move on to 3 marker and 5 marker. There are only few. We are going to end this session really soon, so don't worry. Okay. So name of the drugs, its source and the nature is given in the table below. Find A, B and C. So what is A, B and C? Over here, name of the drug A, source plant is Papaver somniferum. It is a depressant. In Papaver somniferum, the depressant is what? None other than heroin. Now cannabinoids are given. Source plant is what? Source plant, do you know? It is cannabis sativa. Cannabis sativa. Now, what is the effect? It affects on the cardiovascular system. Now, cocaine is given, erythroxylum coca is given. Nature, what does it do? It affects the CNS. It affects the CNS, central nervous system, or dopamine secretion. Dopamine secretion. So basically your answer would be like this. And please mention dopamine secretion also, that will be even more better. Dopamine secretion. Cool. Children, liking the video, do you think you are able to understand things? And let me know in the comment section. And also, don't forget to hit the subscribe button right now. Now, describe any 3 properties of cancerous cells. What are 3 properties of cancerous cells? First one is uncontrolled growth. They will keep on dividing. That is one thing. Second thing, you know, when a normal cell is there, when it divides, suppose from here also 2 cells are dividing. These cells, they will be stopping the division, which is why, because they show something which is known as contact inhibition. They show something which is known as contact inhibition. That means when they come in contact, the growth is inhibited. But over here in this particular case, in the case of cancerous cells, they have lost the property. So loss of contact inhibition is there. There is no contact inhibition that is happening up over here. There is loss of contact inhibition. Always remember. And apart from that, so uncontrolled growth, loss of contact inhibition, and the next thing that is there is what? Metastasis. So from one place they will move on to the next place and they will keep on dividing. This is known as metastasis. Okay, so uncontrolled growth, they divide rapidly without responding to normal regulatory signals. Loss of contact inhibition, they grow even when in contact with other cells, forming what? Tumor. Metastasis, they can spread to distant body parts via the blood or the lymph. These are the properties of the cancerous cells. I told you cancer is super important, please know about them. Now, what are lymphoid organs? Now, write the functions of the following in response to the immune system in our body. So, before I go there, so this is supposed to be last question because we already solved this question, right? So this is going to be last question. Don't worry, we'll make sure it is finished as SAP. And this is for you guys as well, right? So, my dear children, you have bone marrow, thymus, spleen as well as lymph nodes. How does it work? First of all, I'll tell you a brief. So, bone marrow is basically the thing which you have inside the bones. Okay. Now within the bone marrow you have two things. You have red marrow and you have yellow marrow. Yellow marrow is basically responsible for production of fat and also for production of the new bones out there. Red marrow has specific type of stem cells, the blood stem cells, which are called as hematopoietic stem cells. Okay, hematopoietic stem cells, they give rise to the blood cells. So they give rise to different type of cells such as RBC, platelets, and also WBC. In WBC, we know B cells and T cells are also a type of WBCs. B lymphocytes and T lymphocytes are also type of WBCs. B lymphocytes, they mature here itself. In the bone marrow itself, they mature. Okay, they become sensitive to antigen. That means they know that this is the antigen they have to be sensitive to, and this and the things. Okay, now T lymphocytes, they mature in the next organ, which is known as thymus. And since both of these places are the ones where they are getting matured and they're becoming antigen sensitive, I'll together call them as the primary lymphoid organs. Now you have other things also. From here, they are going to go to other places which are secondary lymphoid organs such as spleen and the lymph nodes. So spleen, my dear children, I'll go directly towards there. Spleen is located very near your stomach. This is spleen. And this is actually known as the graveyard of RBC. So red blood cells after 120 days, they die. So they come over here, basically everything is being taken away, only the iron thing is being left. Whatever important is being taken back, rest everything is being broken down in the case of RBC. Now, since spleen has been supplied with the blood which also has RBCs and things like that, they are there here in this particular place, that is spleen. You have lot of WBCs also. Now, suppose a pathogen is coming inside. What will happen? These WBCs are going to act on it and do distribution and kill it. Got it? Which is why spleen is what? A secondary lymphoid organ, because they are affecting over there. Similarly, you have lymph nodes. We have lymph nodes. These round things that you see are known as lymph nodes. So in our body, we have arteries, veins and capillaries. Apart from that, we have a different system which is known as lymphatic system, which carries the fluid which has been leaked from the capillaries. So there are capillaries has certain leaks through which everything, plasma, some types of cells also get released, and there should be someone to collect all of it and give it back to the blood vessel, that is a vein, and it takes to the heart. So that system is your lymphatic system. Now when it takes back, it assures that no pathogen is there. How? Wherever the lymph nodes are there will be having too much amount of WBCs. These WBCs will trap any pathogen which is trying to leak out, which is trying to move around our body. Okay, so lymph node is also going to act like what? Our secondary lymphoid organ. So lymphoid organs where origin, maturation and proliferation of lymphocytes occur. Primary lymphoid organs, bone marrow, thymus, produce antigen-sensitive lymphocytes which migrate to secondary organs to become effector cells. Bone marrow, which is their main site where all blood cells including lymphocytes are formed, provides microenvironment for B lymphocytes to mature and become antigen sensitive. Thymus, primary lymphoid organ for T lymphocyte maturation. Immature T cells become antigen sensitive over here, and later they differentiate into effector cells in other places. Not here. They become effector cells in secondary lymphoid organs. Not here. Now, spleen. Secondary lymphoid organ that filters blood and traps blood-borne antigens, contains lymphocytes and phagocytes that help activate antigen-sensitive cells to effector cells. Same thing. Lymph nodes trap antigens from lymph entering the body, providing sites for lymphocyte antigen interaction. Now, apart from that, ma'am, do we have other secondary lymphoid organs? Yes. In the intestine, we have Peyer's patch, and we also have tonsils over here. Okay. We have tonsils over here. All of them, they are a part of our lymphoid system. Okay, and in your NCERT, mucosa associated lymphoid tissue is also given. That means we have different layers, right? There is a layer which is known as mucosa. Inside that also you have lymphoid tissue. Now there is a percentage given in your NCERT. This much percentage of malt is there? So is acquired by malt. Can you tell me the percentage again in the comment section? So remember, I gave you 3 homeworks. Okay, I gave you 3 homeworks, my dear children. Please go through it and make sure you're writing down the answer in the comment section, because this is going to be super helpful for you. That is why I'm telling you. Okay, done. And with this, we end up everything which was there needed for you guys. If at all there is something else, I'll let you know. And my dear children, remember, on Monday I will be solving all the MCQ questions of the question bank from this chapter, so be there for the live as well. So wishing you all the best. Please take care of yourself. You are going to absolutely rock the examination. Bye bye. See you soon.